Hello friends, I am Dr. Shoeb Ahmad, Assistant Professor in Department of Zoology, Pune College of Arts, Science and Commerce, Pune, Maharashtra. Today we will talk on tissue engineering, the process of tissue engineering, the steps of tissue engineering and what materials are required to perform the tissue engineering and what is the application of tissue engineering. So tissue engineering is the process or technique to regenerate the damaged tissue. Tissue engineering is a branch of medical science or regenerative medicine that deals with the formation of functional three-dimensional tissue by the combination of scaffold cells and the growth factors to achieve the functional living tissue. Through this process, damaged tissue or organ can be regenerated or it can be maintained. So what are the principle, on which principle the tissue engineering technique is being performed? Basically, it involves combination of living cells along with the bioactive molecules with a natural synthetic support that is called as scaffold to build a living three-dimensional tissue, and that would be structurally and mechanically better than the tissue that to be replaced. Means the basic principle of the tissue engineering is the loading of cells and the growth factor over the support system that is called as a scaffold, and then this structure means the scaffold loaded with the cells and the growth factor is called as tissue engineered construct, and this tissue engineered construct is implanted at the site of defect that, and this structure will recreate or regenerate the tissue. The requirement of materials to regenerate the tissue. So there are three materials that is required to recreate or regenerate the tissue through the process of tissue engineering. The first and very crucial requirement is scaffold. Scaffold is a very significant part of the significant material of the tissue engineering, because this is the platform that is used to fill the gap in any organ or to recreate this cell, damaged cell or the tissue. And apart from scaffold, there are two other requirements, that is growth factor and the cells. These are the biological materials, while scaffold is a non-living material. So there are some criteria to achieve effective and long-lasting regeneration of the damaged tissue. These criteria are: adequate number of cells must be produced to fill the defect. Cells must be able to differentiate into desirable shape. Cells must adopt appropriate three-dimensional structural support or scaffold, and it should produce extracellular matrix. Cells must be structurally and mechanically compliant with the native cell. Cell must successfully be able to integrate with the native cell and overcome the risk of immunological rejection, and there should be minimal biological risk. Now, source cell, from where we can get the cell for the regeneration of the tissue. So there are three categories to source the cell. First one is autologous. Autologous cell source means cells can be taken from the patient's own body. Second one is allogenic, means cells can be taken from a human donor. Now third one is genogenic, means cells can be taken from a different animal species. Now cell sources can be further divided into 4 categories, and these 4 categories are mature, that is non-stem cells, adult stem cells or somatic stem cells, embryonic stem cells, totipotent stem cells. So we will see all these 4 cell sources with their examples. First one is mature or non-stem cells. Suppose we have to regenerate the cartilaginous tissue. So for this purpose, we can regenerate the cartilaginous tissue through the mature chondrocytes. Means if we get the mature chondrocytes, we can incorporate mature chondrocytes in the scaffold with its growth factor, and we can regenerate the cartilaginous tissue, for example, ear pinna, ribs, nose, and the joints. But the question is from where we can get the mature chondrocytes. We can get the mature chondrocytes from mesenchymal stem cells. And the source of mesenchymal stem cells are the bone marrow, peripheral blood, and the adipose tissue. Adult stem cells, second type of cell source is adult stem cells. Adult stem cells are undifferentiated cells that stay among the differentiated cell in a tissue or organ of the animal body or human body. These are the body organs that have undifferentiated cells among the differentiated cells. So we can get undifferentiated cell from these organs and we can regenerate the tissue. Embryonic stem cell. Embryonic stem cells are derived from the inner cell mass of blastocyst prior to implantation to the uterine wall. Means if we culture the inner cell mass of embryonic stem cells from the blastocyst, we can get the nervous tissue, we can regenerate the nervous tissue, we can regenerate the skin, we can regenerate the bone, we can regenerate the muscles. Totipotent stem cells. These cells are capable of giving rise to any type of cell. Suppose if we get this totipotent stem cell and we culture the stem cell, that can give rise to any kind of tissue of the body. For example, if we need to regenerate the nervous tissue, we can create the nervous tissue. If we need to create the blood cells, we can create the blood cells. If we need to regenerate the liver tissue, we can recreate the liver tissue. If we need to recreate or regenerate the muscle tissue, then we can do the same. Now, scaffolds. What are the scaffold and what is the use of scaffold? If we see any under construction building, you can observe that at the side of the wall of under construction building, there is a structure made up of bamboo or iron pipes, and this structure is called as scaffold. And what is the function of this scaffold with respect to the under construction building? It provides platform to the workers to supply the materials to construct the building and for their stay during the repairing of the building or during the construction of building. So it is a kind of platform to construct the building. Similarly, With respect to tissue engineering, the scaffolds are also work as a platform to provide materials at the site of the tissue engineering or tissue to be repaired. How scaffold are used? What is the role of scaffold in tissue engineering? Suppose there is a defect in a bone, and that defect is need to be repaired. For that purpose, we need to have scaffold, and this scaffold is incorporated with growth factors and the cells. After the incorporation of cells with and the growth factors, the scaffold will be transplanted to the site of defect, and the scaffold is implanted at the site of defect, and after some time the scaffold carrying the cell and the growth factor will regenerate the missing tissue and it will fill the gap. Materials used to make scaffold. What material we need to have for the preparation of scaffold? Typically, biomaterials are used to prepare scaffold for the tissue regeneration. But the question is, why only biomaterials? Why we can't take any other materials? Why we can't take metals? So first you should need what is biomaterial. Biomaterials are non-living materials that is used in medical device. And biomaterials are used to make scaffold because of its biocompatibility and biodegradability. Means biomaterials are used to make scaffold because of its ability to degrade inside the recipient's body by the body's own system. It do not create any kind of inflammatory or immunological reaction inside the recipient body. There are three groups of biomaterials that are used in the formation of scaffold for the tissue engineering, and these are ceramics, synthetic polymers, and the natural polymers. If you see, under the category of ceramics, hydroxyapatite and tricalcium phosphate are used to make the scaffold for the regeneration of bone. Synthetic polymers, under this category, polystyrene and polyglycolic acid are used to make the scaffold for the regeneration of blood vessels and tubular structure. Now, third one is natural polymers. The best example of natural polymers are silk protein that is called as silk fibroin and blends of collagen. These two proteins are used to make scaffold for the regeneration of cartilage and the bone. Mechanical properties of scaffold. What mechanical properties should be there inside the scaffold? Scaffold should be consistent with the anatomical site into which it is to be implanted. Means it should not move out, it should not bulge out from the site of implantation. It must be strong enough to allow surgical handling during the implantation. Biocompatibility of scaffold. This is very first criteria of scaffold that is used in tissue engineering, that it should be biocompatible and it should allow the cells to adhere. Means it should allow the cells to adhere, to join, to combine with each other and to proliferate, to multiply, and it should allow these cells to function normally, and it should also allow the cells to migrate onto the surface of scaffold. And the very important point is that after implantation, the scaffold should not elicit any immune reaction, any kind of inflammation, any kind of rejection. Scaffold or the tissue engineer construct are not a permanent implant. Means the scaffold along with the cell and the bioactive molecule is not a permanent implant inside the body of recipient. So therefore, the scaffold must be biodegradable. After implantation, as soon as the cells start multiply and start the formation of living and three-dimensional tissue, the scaffold should be degrade inside the body of recipient by the body's own system, and the byproduct of this degradation should be non-toxic and should be exit from the body without any hurdle. Now, tissue engineer construct. What is tissue engineered construct? When cells and bioactive molecules are loaded on scaffold, the structure formed is called as tissue engineered construct. And this tissue engineered construct is now given a shape on the basis of tissue to be re-engineered, tissue to be engineered or to be recreated. And this tissue engineered construct is implanted at the site of regeneration. Architecture of the scaffold. On the basis of tissue to be regenerated, scaffold produced in different designs and shapes from a variety of biomaterials by using the fabrication technique. Means the shape and the design of the scaffold depends on the shape of the tissue that is to be repaired. Scaffold should have interconnected pores and it should have enough porosity to ensure cellular migration, cellular penetration, diffusion of nutrients within the tissue engineered construct and to the extracellular matrix. It means the scaffold should have interconnected pores to allow the migration of cells, to allow the diffusion of nutrients. So process of tissue engineering means what are the steps of tissue engineering? The first step of tissue engineering is to prepare the scaffold on the basis of tissue to be re-engineered. The material is to be chosen and the scaffold is prepared. Now shape scaffold as needed on the basis of tissue to be, on the basis of the shape of the tissue to be recreated, recreation, the scaffold should be given shape accordingly. Now the scaffold is incorporated with the cells and the growth factors. Now after seeding the cells and the growth factor, the cell multiply and grow in three-dimensional tissue. Means as soon as the scaffold is incorporated with the cell and the growth factor, the cell and the growth factor starts to create the three-dimensional tissue. Now the tissue engineered construct is implanted inside the body of recipient. Now after implantation of tissue engineered constructs inside the body of recipient, the cell recreate their intended function. Now the blood vessels attach themselves to new tissue, means the blood vessels of the recipient body starts to attach themselves to the new tissue. Now scaffold dissolves as soon as the cell multiply and there is a formation of three-dimensional functional tissue and blood vessels starts to attach themselves to the new tissue. The duty or the function of the scaffold is now ended, and now it should be dissolved by the body's own system. The scaffold is now dissolved by the lytic enzymes of the body. Now there will be formation of three-dimensional tissue and it will fill the gap inside the organ and form the three-dimensional functional tissue. So this is about the tissue engineering, steps in tissue engineering, techniques in tissue engineering. I hope you understand the things, you understand the techniques and the steps in tissue engineering. So thank you so much. Thank you, Sanjay. Thank you so much.